Hello and welcome back to our course Data Analysis in Astronomy and Physics, Lecture 1. In this lecture I would like to give a short introduction to basics of data itself. We will talk about sampling and I will present a few different kinds of scientific data formats that are commonly used and discuss their advantages and their disadvantages. When we talk about data, it is common to distinguish between observations, sometimes also called cases, and variables. To explain the difference, I show here a simple data set. This is the passenger list of the Titanic, and it shows per passenger a number of different entries such that each row corresponds to one passenger. This means each observation corresponds to all the data that belongs to one passenger, and this is a collection of different variables measured for this observation. An astronomical example would be that an observation could be, for example, a star, and different variables that we measure corresponding to the stars are, for example, their mass, their luminosity, their distance, their spectral class, and so on. Most of the time we are interested in relationship between variables. So it's not uncommon that we observe variables of an object and we observe a certain relationship. If these variables are connected with one another, this is called association. They are dependent. An association can be positive or negative, and we say that two variables are not, if they are not associated, they are independent. Here on these two figures I give two examples. On the left hand side I plot the mass-luminosity relation of the nearest 100 stars. The mass is given on the x-axis, the luminosity on the y-axis, and we observe a very clear relationship with a couple of outliers below the trend line. On the right hand side I plot the radiative decay of sodium-24. The x-axis is the measured time in hours and the y-axis is the measured counts per second. Variables come in different kinds. There are numerical, quantitative variables. They can be either continuous or discrete. So for example, a continuous quantity could be the height of a person. A discrete quantity could be, for example, the number of children this person has. And there are qualitative variables or categorical variables. These can be either regular categorical variables. For example, looking at the case of a person, that could be the sex, so either male, female, or others. Or it can also be an ordinal variable. These are qualitative variables that have some internal ordering, so that it's clear that you can put them in a regular ordered way. Another important term is sample, and with sample the important quantity is the size of a sample. This is because usually we don't have access to the full population of items or entities that we are interested in. That means if we want to study them, we have to study a sample, so a subset of this population. And the size of the sample is an important factor that determines the quality of our study. A good example is that if our sample size is limited, or, for example, suffers from certain bias, for example, look at this example here of early smoking research looking for negative health effects of smoking. These studies also suffered from low sample size and gave, for example, anecdotal evidence of persons in the study that smoked a lot and never had any health problems. But of course, this is only an anecdotal evidence for a single individual person and usually cannot be generalized for the whole population. So it's important to have a representative sample. This is not necessarily easy, particularly in astronomy. Here I give you an example. This is a study looking at planets that have been found using the Kepler satellite. The study looks at multi-planet systems and tries to study them and connects them to the best studied planetary system that we know, the solar system. So the solar system is compared to the Kepler data, but of course the population that we are interested in is all the planets in the Milky Way. So our goal is to understand the fundamental physics of the star and planet formation and the fundamental properties of all the planets in the Milky Way. And to derive them, we have access to a small sample. And the study, for example, finds that planets in the same system tend to be of the same size, and that the orbital distance between the first pair of planets seems to be a good predictor of the orbital distance of the third planet and the fourth, and so on. Connecting this to the solar system shows that the solar system seems to be different from the Kepler sample. So, for example, looking at the planet size, we see that the solar system has some differences in the individual planet sizes. So this could have a couple of reasons. For example, that the Kepler sample is not necessarily representative for all, or that the solar system is not a representative planetary system. When we perform studies, statistical studies, there are two different classes of studies. There are observational and experimental studies. The observational study is collecting data such that it does not directly interfere of how the data arises. Astronomy is a very good example of a research field that commonly does observational studies, because we have no way to set up an interstellar experiment. We have no way of affecting the properties or the collection of stars that we would like to study. We can only observe the universe as it is. The disadvantage of these kind of studies is that it's only possible to establish an association. It's not possible to, from these statistical studies, establish causal connections, and we will come to that in a minute. There are also then studies that uses past data, retrospective studies, and prospective studies. These are studies that continuously keep collecting data throughout the whole research. During experiments, particularly in social sciences and medical studies, you usually randomly assign test subject to a certain treatment and compare the results to control groups that have not been subject to this treatment, and this is done to establish causal connections. Again, in astronomy this is very difficult. In other physical fields usually you can set up experiments. So this is again a view graph showing the difference between the two. Here the underlying example is we might be interested in how does regularly working out change or affect the average energy level of a person, the average calories consumption. And if we do an observational study, we could, for example, measure the energy level of people that regularly work out and compare them with a group of people that doesn't work out. The problem is that there might be some additional factors that determine whether people work out or don't work out at all, and other influences that might play into the results here. So it's better to set up an experiment that you take up an representative group of persons, randomly assign them to a group of people that are supposed to work out and the others that don't work out, and then compare their energy levels. Okay, as I said, we are interested in correlations, in associations between variables, and particularly interested in a causation and in a relationship between two variables. These two variables, the explanatory and the response variable, are said to be causal if the change in the explanatory variable actually causes a change in the response variable. And here the explanatory variable is usually shown on the x-axis, and it's the independent variable, while the response variable is the dependent variable, usually shown on the y-axis. The difference between the two shows that between association and correlation, both describing a relationship that exists between two or more variables, and if we are describing the quantitative strength of a relationship, then we usually use the word correlation. So when we are talking about numerical variables, for example. If we observe an association, then there are possible explanations for these observed association. It could be that the explanatory variable is actually causing a change in the response variable. The response variable could actually cause a change in the explanatory variable, so that we exchange cause and effect between the two. We could have the case of confounding, that is, that there may be causation, but there are too many uncontrolled variables that also play a role. And there is the possibility to have a common response or a coincidence, which means that there is no actual causation and that the association between the variables can be explained by other variables associated with both the explanatory and the response variables. So looking at the figure in the bottom, where we show a clear association between the per capita cheese consumption as a function of time and the number of people who died by becoming entangled in their bed sheets as a function of time, what do you think? As I already said, sampling means selecting a certain subset of entities from the population and then trying to derive properties of the whole population. There are different kinds of sampling, and depending on what method of sampling you use, you might introduce certain bias into your study. The most general sampling is the so-called census, which means we sample the entire population. This might be very difficult because some members of a population might be more difficult to measure than others. And so very often it's impossible or at least very impractical. This can be either because our resources are limited, so we might have only a limited time to perform the study, or manpower, money, and so on, or technology, which is usually the limiting factor in astronomical studies. It might be that not all members are accessible, for example in astronomy, because of physical limitations. The visible universe is smaller than the entire universe. So we cannot sample the entire universe. And of course it might be that our populations, for example, are not static and evolve or change during our research. So usually it comes down to sampling a subset, so measuring a representative sample of individuals. Some of the biased sampling strategies are, for example, a convenience sample, which means we only choose those members of the population that we have easy access to. For example, only look at the most nearby stars, because these are the ones that I can study with a cheap telescope. You can also sample from a very specific area, which means that this limits the applicability of our study to that sampled area. This is an extreme form of the biased sampling because some members of the population are totally excluded. So for example, we could look at the population on Earth, but only on those members that have, for example, a shoe size between 40 and 44, and then derive some properties or study this group. And it's very difficult to generalize the results then to everybody. There is a self-selection bias in social sciences, and that's when the subjects of our study can control whether they are actually part of the study or not. So that's something that in astronomy or in physics is a rare case. It might be that we have a pre-screening of our trial participants or of the possible sample candidates, and then select only of these pre-screened ones. So this also introduces a certain bias. An astronomical example would be that we only look at the most luminous objects in the universe and then do statistics on them. There are others, and I mention a few here on this slide, but I think everybody gets the general idea. It's very important when you select a sample to make sure that it is as representative as possible, and during the research, when you conclude, keep in mind to conclude general properties of the population. For this reason, usually there is a random element involved in sampling, but even here it's possible to apply different random sampling strategies. So there is simple random sampling, such that we just randomly select members of the population and study those. There's also the possibility to do systematic sampling. One example could be, for example, to look at catalog entries. For example, look at stellar catalogs listing all the stars that's been characterized so far, and then take every fifth entry. Depending on the way the catalog is arranged, this should also be a relatively representative sample. You can also do stratified sampling, which means that you split your population into certain categories and then sample from these categories individually to make sure that every category is represented in your samples. Of course, you have to make sure that the numbers that you draw from each category is representative to the relative occurrences of these categories in the population. And then there is cluster sampling, which selects samples from groups, from subgroups in the population. For example, an astronomical example again would be that we look at all stars from 5 random globular clusters and study those. Again, here in all these different strategies, it's always important to check how much this hampers the ability to draw general conclusions from your samples. And this view graph again tries to show the different strategies that I just presented. Okay, just to summarize, to have as general results from our study, it's important to perform random sampling and to minimize any bias in how members of our sample are selected, and to draw or to find causal effects. It's important to then randomly assign members from our sample to a treatment group and to a control group to actually distinguish between causal and non-causal effects. And the table here on the bottom gives you a brief overview how these different strategies, random assignment and random sampling, then allows you to get causal and generalizable results.